Traditional methods of isolation and enumeration of bacterial colonies, the pour plate method. Established in the late 19th century by Robert Koch, a German physician and a founder of bacteriology, a branch of microbiology, the pour plate method is still widely used in isolating and enumerating pure cultures of microorganisms. Koch is most widely known for using this technique, among others, for identifying the bacteria responsible for anthrax, tuberculosis, and cholera. This method involves mixing a liquid sample with molten agar and then pouring the mixture into a petri dish. Some uses include environmental monitoring, clinical diagnostics, microbial contamination in food, and pharmaceutical testing. The procedure for preparing pour plates is pretty simple. Samples are diluted to ensure colonies can be countable. Agar is prepared at 45 to 50 degrees Celsius. The key is to keep the medium liquid, but not so hot as to kill the microorganisms. Molten agar is then poured over the sample within a petri dish and is gently swirled together. This is then solidified at room temperature, and the plate is then incubated upside down. After incubation, colony-forming units can then be calculated. Some advantages of the pour plate method are the enumeration of viable cells or colony-forming units within a sample, the growth of aerobic or oxygen-dependent organisms, anaerobic organisms which are not oxygen-dependent, and facultative organisms which can reproduce with or without oxygen. Another advantage includes the isolation of these colonies from mixed samples. It's also suitable for heat-sensitive organisms, and it's fairly easy to perform. Some disadvantages to this method is that it is more time-consuming than other methods. Serial dilution is necessary for the samples, and you must wait for the medium to solidify. This method also requires precise temperature control, or the microorganisms within the sample may be killed. Solid or semi-solid samples must be suspended prior to inoculation, which can be tricky if they are not easily soluble. Also, more time is needed for cell growth, as anaerobic organisms tend to grow more slowly as their energy production is less efficient than aerobic organisms. It's very important to keep these pros and cons in mind when considering the most advantageous methods for your research, and hopefully this video has provided some useful insights. If you have any more questions, visit the references at the end of this video, and thanks for watching.